In the last video, we talked about how cancer can arise from abnormalities in cancer-regulating genes. These genes fall into two major categories: oncogenes, which promote cancer, and tumor suppressor genes, which inhibit it. There are many genes in both groups. Some well-known oncogenes include SRC, HER2, RAS, EGFR, and BCL2. Common tumor suppressor genes include p53, BRCA, RBP10, and BEX. Among these, we already discussed the SRC in the previous video. So in this one, we'll focus on HER2, which plays a key role in cancer development, especially in breast cancer. Our body controls cell growth very precisely. When certain cells need to grow, the body sends a signal called a growth factor. This binds to a growth factor receptor on the cell surface. Once bound, the receptor is activated, send a signal inside the cell to trigger cell division and growth. There are many types of growth factor receptors, and HER2 is one of them. A unique characteristic of HER2 is that it can become overexpressed due to abnormalities in gene regulation. This leads to excessive growth signaling, ultimately turning a normal cell into a cancer cell. HER2 overexpression is particularly common in breast epithelial cells, which is why HER2 is considered a major oncogene in breast cancer. However, not all breast cancers are caused by HER2. So breast cancers are generally divided into two types: HER2 positive, which show high HER2 levels, and HER2 negative, which do not. HER2 negative breast cancers can be further classified into hormone receptor positive and triple negative types. But in this video, we will focus only on HER2 positive breast cancer, which accounts for about 25% of all breast cancers. Since HER2 drives cancer growth, scientists wondered, what if we block HER2? Can we stop the cancer? This leads to the development of a targeted therapy, a treatment that specifically attacks molecules like HER2. Researchers developed antibodies that attach only to HER2, and when tested, they worked. Tumor shrank. Among these, the most famous is trastuzumab, approved by the US FDA in 1998. Its brand name is Herceptin, and is still widely used today. In fact, Herceptin was the first antibody drug ever approved for cancer treatment. Trastuzumab fights against cancer in several ways, and I will explain the three main mechanisms. First, it blocks HER2's function. By binding to HER2, the antibody prevents it from sending growth signals into the cell. It also reduces the number of HER2 on the cell surface. Once trastuzumab binds to HER2, it triggers endocytosis, pulling HER2 into the cell. There, it is destroyed in the lysosome, the cell's recycling system. With a fewer HER2 on the surface, the growth signal weakens. Another action of trastuzumab is that it helps immune cells attack cancer. One of these immune cells is the natural killer, or NK cell, which has a surface molecule called FcR that binds strongly to antibodies. So, when an NK cell encounters a cancer cell coated with trastuzumab, it recognizes the cancer cell through the binding of FcR to the antibody. Then the NK cell kills the cancer cell by releasing toxic molecules. This mechanism is called antibody-dependent cellular cytotoxicity, or ADCC. In short, trastuzumab works in three ways. It blocks HER2 signals, removes HER2 from the surface, and helps immune cells attack cancer. In this video, I explained how the HER2 gene contributes to breast cancer and how targeting HER2 can lead to effective treatment. I hope this helps you better understand HER2-positive breast cancer and its therapies. Thank you for watching.